Hello, everybody. Welcome to Vigene's presentation. My name is Jeff Hung. I'm the General Manager for Vigene Biosciences, a Charles River company. The title of my presentation is Scalable GMP Production of AV and AV Reference Materials. And first of all, I would like to acknowledge Alliance for Regenerative Medicine for organizing this cell and gene therapy meeting on the Mesa. I look forward to meeting a lot of you here in Carlsbad this week. On June 29th, 2021, Vigene becomes part of the Charles River company, which is a public traded company. This is a disclaimer. AV has become a workhorse vehicle for delivering gene therapy in vivo and into cells for cell therapy. The benefit of AV can be summarized as on this slide. I coined the 5S. It's small and it's very tough, 18 nanometer with no envelope. With one contig, the gene codes 3 viral particle proteins, BP1, 2, 3, performing different functions required for viral vector, including transduction. The third one is safe. The recombinant AAV does not have replication competency, so it's very safe. And this one also caused a lot of difficulty in manufacturing. The fourth one is it has serotype-based tissue specificity and rendering the gene therapy to be very targeted for different organs and tissues. The fifth, but not the least, is the systematic approach. Regardless of the foreign DNA sequences, most of the genes can be packaged at ease when the payload is below 4.7 kb capacity. So AV has become a mainstream gene delivery vehicle, but the manufacturing for AV has been a bottleneck for gene therapy accessibility and affordability. And over the last decade, Vigene, as well as many other companies, has developed the technology for AV scale-up to meet the challenge to supply gene therapy around the world. And for those who don't know Vigene, we have 17 years of GMP viral vector experience and development experience. We have 110 square feet new lab and office space, including 62,000 GMP space. In the context of today's presentation, we have a lot of experience with scaling up from 10 liter all the way to 500 liter. We're now in the process of establishing 2,000 liter suspension bioreactors. In addition to fully GMP viral vector production, we also have full GMP plasmid production. And we are a fully integrated service CDMO from research production all the way to GMP commercial. And borrowing our experience of scaling up, I would like to highlight 3 aspects of success factors in scale up. Well, one aspect is equipment, the other one is facility, and the process will be the focus of the rest of the presentation. For equipment, one needs to take care of the temperature control, mixing, pH control, and process parameters such as DO, CO2 metabolite. And for facility, material storage, cold chain, material flow, EMQC changeover, and HVAC air change are pretty critical to achieve the success. The AV production process scale up is not that easy. So it has a lot of ramification on different aspects of the bioprocessing aspect. I would like to highlight 4 aspects of AV scale up, including cell line, upstream scale up, downstream scale up, and the plasmid production scale up. To understand the AV scale up production, we have to understand the three major manufacturing platforms for AV packaging. The first one is a helper virus, including HSV-based helper or adenovirus-based helper. The second one includes SF9 and baculovirus-based AV packaging. Last one is triple transfection, and that's a helper virus-free. And so this one has become a major success story for both commercially approved gene therapy and also a lot of clinical material gene therapy. So I'm going to focus on the rest of my presentation on triple transfection plasmid-based AV packaging. And by and large, there are adherent or suspension-based AV packaging. And for Vigene, we have a lot of experience with suspension-based scale up. And so first, the line of data for the scale up come from the cell line optimization, which I will touch upon first. As you can imagine, the scale up for cell line is very critical. And if the cell line have a high productivity, the overall yield will be much higher than otherwise. So Vigene has developed the proprietary HEC293 cell lines for AV packaging. So on this slide, we represented the productivity data in comparison with a reference HEC293 suspension cell line on the market. And so this serotype, we can produce 4.2 E13 purified AV and using a Vigene proprietary cell line, whereas the other cell line is 1.8 E13. So this achieved the 2.5 fold productivity. And so the serotype on this one is AV6. And in addition to cell line, I think I'm going to now switch to the upstream, that is the plasmid construction. So the plasmid, in addition to P helper, both the gene of interest and the RepCap plasmid can be engineered to increase productivity. Here is one patent that we have received for RepCap plasmid construction and to increase AV2. And for AV2, as you can see on this slide, and we can achieve as much as 10x productivity using a Vigene AV2 RepCap technology. And then the other aspect for triple transfection based AV production is transfection step. There's a lot of different transfection agents available, and one can also optimize the transfection time in addition to PEI DNA ratio. So Vigene spent a lot of time optimizing the PEI DNA ratio versus, and also the transfection time. So this one represented some of the results that from our optimization. So the optimized protocol can increase AV productivity as much as 20x. And downstream scale up is also very critical. A lot of AV are wasted in the downstream process. So the recovery rate for a lot of processes is not very high. So the scale up consideration, including cell disruption, mechanical or detergent disruption to release AV from the cell, and benzonase digestion to get rid of the whole cell DNA, and the rest of the plasmid to release the AV from this whole cell environment. Clarification, resin affinity resin, and ion exchange chromatography to enrich full capsids. And the final formulation is also very critical. And for full capsid enrichment, ultracentrifugation during the scale-up process is often not very friendly for large scale. So there is a pretty urgent need to replace ultracentrifugation using chromatography-based full capsid enrichment. And the Vigene has developed a good empty full enrichment using chromatography. And as you know, the AV capsids, they can package viral particles independent of payload. Even without any DNA content, empty capsid can be formed. And fragmented payload can also be packaged into capsid. So it's of critical importance to separate the full capsid from the empty or fragmented payload. The reason is twofold. One is empty or fragmented, they don't have any potency. And secondarily, they can also elicit immunogenicity that is not desired. So for gene therapy program, it's of great importance to achieve consistent full capsid ratio and to enrich full capsid in the gene therapy product. So on this slide, we showed some of the result to separate the full capsid from empty. And the top is a chromatogram and using AEX. And then the lower panel represents the TEM data showing the enrichment of a full capsid. The enrichment is shown in even colored dots on the bottom. And the one that circled by red, they have a dark core. That's the empty capsid. The red is artificial color. So we can enrich as far as 89% full using AEX. So it's a fully commercial production ready method for downstream. And last but not least, plasmid production scale up. So as you can imagine, plasmid scale up is very important for both the quantity and the quality of AV production because they serve as the starting material for AD packaging. And the Vigene spent quite a bit of time to optimize a plasmid manufacturing process, including addressing the challenges for low productivity, high whole cell DNA, and get rid of contaminating bands and low supercoil homogeneity. And by going through the plasmid design, the downstream process to get rid of host cell DNA, and then get rid of contaminating bands, and using downstream process to achieve supercoil homogeneity. So I have gone through the manufacturing bottlenecks in the AV packaging process, including plasmid, upstream, and downstream production. Now I'm going to switch gear to address another bottleneck in the AV manufacturing and analytics, that is AV reference materials. And up to 2020, there were only two kinds of commercially available AV reference materials, and both are from ATCC. They were produced by AV reference standard working groups back 10 years ago. And after those working groups manufactured the AV reference material, they deposit at ATCC for global distribution. And those batches are running low. In addition, some of the titers are relatively low and making them not compatible for certain assays. For instance, AV2 reference is only 3.2 E10 viral genome per mil, making it incompatible with analytical ultracentrifugation method. And the third drawbacks is that some of the other serotypes are not covered in addition to AV2 and AV8. And the last one is there's no empty full capsid ratio analyzed using those AV reference material. So to meet those challenges, Vigene set out and spent almost a year to develop AV reference materials for 6 serotypes, which are listed on this slide. And so we have gone through the rigor like GMP production to produce those 12 batches of AV reference materials. using triple transfection and using aldexanol ultracentrifugation to enrich full capsid and separate the empty from that. And those reference materials gone through rigorous QC, which I'm going to present the data for. So altogether, 12 batches of AV reference materials are produced, 6 full capsid reference materials and 6 empty capsid materials. The empty full capsid data analyzed by transmission electron microscopy are captured on this slide. The full capsid ratio for full capsid reference range from 56% all the way to 82%, whereas the empty percentage for empty capsid reference material range from 84% all the way to 99.5%. So those AV reference materials, 12 batches, are analyzed using rigorous QC, close to a dozen different assay methods. Other than the TEM, all other methods are GMP qualified. So including qPCR, ELISA, bell burden, mycoplasma, silver stain, endotoxin, and the other aspects. So on the right-hand side on this slide, you can see the certificate analysis is represented using presenting both TEM data and also the silver stain data. The AUC data are not available on the CFA, but we did partner with Beckman Coulter to generate AUC data for this 12 batches of materials. The data are presented in this slide. And so this slide summarizes the comparative data of empty full capsid using 3 methods, AUC, TEM, and qPCR ELISA. The left panel represents the data for full capsid and analyzed by TEM, AUC, and the qPCR versus ELISA respectively. And the empty capsids percentile are analyzed by TEM and AUC only. And the qPCR, because they don't have any payload, is not possible to do that qPCR ELISA assay. So one conclusion we can draw from this comparative data is that the AUC, TEM are highly correlated. And so when the AUC and the TEM call a certain reference material full or empty, the other methods also have the right call. The discrepancy, however, between qPCR ELISA and the TEM AUC is a little bit bigger than AUC versus TEM. For instance, some of the qPCR ELISA give 104% full ratio and 171% of the ratio. The reason for those discrepancies could be due to the very fact that those are derivative to a number of 2 independent assays, and which both have its CV and variation. And from this comparative study, we feel that the empty full ratio are pretty well analyzed on this 12 batches of reference materials. In addition, we also feel AUC have a little bit better resolution and give a more definitive call for full capsid, and based on the chromatogram of the AUC final data analysis. So obviously, for gene therapy and AV production testing, there are still many other obstacles. And this roadmap is something that I wrote down in terms of the sum of the bottlenecks we have to overcome as industry. And I would like to finish with this slide and regarding Charles River. So Charles River has become a major player in the CDMO service for serving cell and gene therapy workflow. from HemoCare Celero of local phoresis cell collection to cell therapy production offered by Cognate, and to viral vector and the plasmid production offered by Vigene and Cognate, form a end-to-end solution for the cell and gene therapy companies. And so we have quite a few representative and executive from different companies here in Carlsbad attending the cell and gene therapy meeting on the Mesa. So we look forward to meeting you this week. And now I'm going to address any questions that you may have.